In this video, I'm going to be talking to you about vaccination. I'm going to be talking to you about what vaccination is, how they are developed, what makes an ideal vaccine, and why vaccination is so important. Before we start on vaccinations, I just want to spend a minute or two with a couple of key definitions about the different types of immunity. So, immunity can be split into some different categories. We can talk about natural immunity and artificial immunity, and we can talk about passive immunity and active immunity. So, natural active immunity is your immune system. It's your T cells and your B cells and your phagocytes and the production of antibodies and the production of memory cells and all that stuff. That's active natural immunity. Passive natural immunity is immunity which is still natural. So it's not an artificial process, but you personally are not producing any antibodies. You are not producing any memory cells. So whereas natural active immunity is a long-term thing, you've developed immunity and those memory cells will be in your bloodstream for a long time, and you'll be able to incite a secondary immune response if you are introduced to that pathogen again at a later date. Natural passive immunity is short-term. The most common examples of this are the presence of antibodies in the mother's milk. So it's really all mother-to-baby transmission of antibodies. So when babies are first born, their immune system is not fully developed yet, and they could not fight off most infections. So because, of course, the pathogens that the baby and the mother are being exposed to are the same, the mother makes those antibodies and passes them into the baby through the mother's milk. Now, this is really beneficial to the baby because it helps them, but it's only a short-term process. Those antibodies are proteins and they will eventually be broken down within the baby's body, but it's enough to keep them going until their own immune system kicks in, but it's a short-term thing. And remember, before the baby is even born, when it's a fetus, the mother's antibodies pass across the placenta into the fetus to protect them while they're still in utero. When you first give birth, you produce a substance called colostrum. It's basically your first breast milk. This contains lots of antibodies, and it's really important for protecting the child in their very first few days of life. So we have the mother's antibodies passing across the placenta to the fetus in utero. We have colostrum, which is a highly concentrated fluid containing lots of antibodies. And then we have antibodies in the mother's milk. These are all examples of natural passive immunity. So then we have artificial immunity. So passive artificial immunity is when you are injected with some antibodies. So imagine that you have got something wrong with you where a very rapid immune response is needed. So for instance, you've got tetanus, or you just got bitten by a really venomous snake or a terrible spider and you're going to die very quickly. So antibodies are injected into you. These are called humanized antibodies. So say you had tetanus, you would be injected with the tetanus antitoxin antibodies, because remember the reason tetanus makes us sick is that it produces this toxin that causes these spasms in our muscles. These antibodies need to be humanized. This is usually because they're produced in animals. So for instance, we might inject a horse with the tetanus toxin, and then the horse's immune system would kick in and produce the antibodies and the plasma cells and the memory cells and all that sort of thing. And over time, we would keep injecting them with the tetanus toxin, and they would get a greater and greater concentration of the tetanus toxin antitoxin antibodies in their bloodstream, and then we can extract those. We would need to chemically modify them to humanize them so that our immune system will respond to them. And then we would inject those into somebody that has been infected with tetanus. So remember that this only provides a short-term protection, because these antibodies that you've been injected with will be destroyed both by phagocytes in the spleen and in the liver. So these antibodies are proteins, and they will eventually be destroyed by your own immune system and your own body. So it's a short-term protection, but it's very rapid because you don't have to go through the process of producing your own antibodies. And then the final one is active artificial immunity. Active artificial immunity is vaccination. What is a vaccine? A vaccine is a preparation which contains either antigens from a particular pathogen or a dead or weakened version of the microorganism. So it could be weakened by being heat treated or treated in acid, or it could be completely killed. This will then be injected into your body, and it stimulates the production of antibodies. So it triggers your T cells and your B cells, triggers your B cells to differentiate into plasma B cells to start to produce and release antibodies into the bloodstream, and it also triggers the formation of memory cells against that particular microorganism. This is a long-term protection because those memory cells will remain in your bloodstream for a long time. And if you are exposed to that pathogen again, so if you're exposed to an active version of that pathogen, if you become infected with it, those memory cells will very rapidly differentiate into plasma B cells and start to produce the correct antibodies. So you will essentially show the secondary immune response. Sometimes people need booster shots of vaccinations. This helps to maintain higher levels of antibodies within the bloodstream so that you maintain a longer term immunity. So we mentioned earlier a few ways that we could introduce antigens into our body to activate our naive lymphocytes to divide without actually making you sick. So we talked about a live attenuated or weakened version. We talked about isolated antigens, so not actually injecting the cell, just the antigens from the surface. We talked about dead microorganisms. You could directly inject the toxin, but a modified version of that toxin, or you could genetically engineer the microorganism to no longer be pathogenic. Vaccines are often injected. This is really because often, if they are taken orally, they may be broken down by the enzymes of your digestive system, or they may not be able to be absorbed into the bloodstream because the molecules might be too large to pass across the lining of your stomach or the lining of your small intestine. So when developing a vaccine, there are a number of questions that we have to answer. So there are a number of problems or challenges that we have to overcome. So one of the first things that we have to consider is what sorts of preparation are we going to use? So are we going to kill the microorganism? Are we going to inactivate it by chemicals or radiation? Now, the benefits of that is that it's much more stable as a vaccine. You don't need to put it in the fridge, so it's much easier to store and transport. This is really important to consider because if we want to have a successful vaccination program, we need to be able to store and transport this vaccine large distances and maybe to really rural areas. When you inject dead vaccines, these are usually safer because they cannot revert to the virulent strain, so it's less likely somebody will get sick from the vaccine. But one of the problems with this is they're normally only weakly immunogenic, i.e. they won't, with one shot, you won't get full long-term immunity, and you tend to need several doses or boosters. The problem with that is uptake. So how do you ensure that people return for their next appointments? How do you ensure that this program is going to be successful? It's really difficult. Also, when you use a dead vaccine, it stimulates mainly humoral immunity, so it requires adjuvants. Adjuvants are chemicals that stimulate the immune system to respond. So these are extra chemicals that need to be put into the vaccine. If you attenuate it so it's alive but it's non-virulent, you basically grow the microbe in lab conditions that are adverse. So you remove the virulence. So there are mutations that remove virulence. Because it's alive, it elicits a much stronger cellular and humoral immune response. So it can often confer lifelong immunity with only 1 or 2 doses. But one downside of using an attenuated vaccine is that there is the remote possibility of reversion to its virulent form. So there could be a mutation and it becomes dangerous again. Also, it's less stable than a dead version, so it requires refrigeration, which adds expense to any vaccination program. In addition to that, another problem is it can't be given to people that are immunocompromised, so it reduces the number of people that actually have access to the vaccine. So if we were trying to create an ideal vaccine, what would we want? We would want it to cause your body to mimic the natural response. So for it to induce a cellular immune response and cause the production of antibodies and the production of memory cells. We would expect it to elicit antibodies against all strains of that particular pathogen. We would expect it to be long lasting. We would expect it to be cheap. We would expect it to be stable, so the pathogen wouldn't mutate and become virulent again. We would want it to have minimal side effects. We can't really say no side effects, okay, because there's always going to be some side effects. Like, that's just an inevitability to introducing something into your body. We just need to determine an acceptable level of side effects. We need it to be easy to administer, and we want it to be easy to store and transport. Essentially, we want the vaccine to be economically available in large enough amounts to elicit herd immunity. And let's talk about what that word herd immunity means. Herd immunity is a form of indirect protection from infectious disease which occurs when a sufficient percentage of a population has become immune to an infection, either through vaccination or through previous infections, thereby reducing the likelihood of infection for individuals who lack immunity. This is because the immune individuals are unlikely to contribute to disease transmission. This disrupts the chain of infection and stops or slows the spread of disease. The greater the proportion of immune individuals in a community, the smaller the probability that non-immune individuals will come into contact with an infected individual. So this is why when we have a vaccination program, there will be a target as to what percentage of the population needs to be vaccinated for it to be successful. Because if the number vaccinated is below that level, you won't develop herd immunity and you aren't going to protect the population. This number differs for different pathogens. This number is known as the HIT, or herd immunity threshold. The main thing that determines the HIT is how infectious a particular pathogen is, i.e., the average number of new infections caused by each case in an entirely susceptible population, so in a population where no one is immune. So if we think about something like measles, which is an airborne infection, it has an RO value, so the number of new cases caused by one infected person, of between 12 and 18. That's huge. That means that one person with measles going out into a society where everyone is susceptible to measles could infect up to 18 other people. This means that a successful vaccination program for measles would need to vaccinate around 95% of the population. Let's compare that to something like mumps. Mumps is an airborne droplet infection, and the RO value for mumps is between 4 and 7. So the herd immunity threshold for a vaccination program for mumps is more like 85%. Flu is much less infectious. Flu has an RO value of about 1.5 to 1.8, and you only need to vaccinate around 35 to 44% of the population in order for the vaccination program to be successful. So there are a number of factors that determine how many people in a population that you need to vaccinate in order for that vaccination program to be successful. So in this video, we have covered the different types of immunity, what a vaccine is, what could be present in a vaccine, the pros and cons of different types of vaccine, and what makes an ideal vaccination program. So, although you might not be expected to be able to recall and regurgitate all of this information, what this information is useful for is to apply. So if you were given an example of a vaccine, you would need to apply these things that you now know about vaccines to decide or evaluate on the potential success of a particular vaccine. So this information is useful for you to be able to apply to potentially unseen scenarios. But there's quite a lot for you to get your head around. And one thing that you really do need to learn, because it's a really common 2-mark question, is the definition for a vaccine. All right, I think we'll leave it there for now. Well done.